University of Montana, Tyler. Tyler is going to talk to us about all the threats we face. And this will be a very interesting presentation for us, for me and everybody. Because the threats we face include phosphates, right? From fertilizer, includes smallmouth bass, right? Because that's a vociferous fish that eats the food, the nutrients of fish that we want in our lake that are more natural. So there's there's more. I could cover more, but I don't want to take too much time. But if you will, there's a haunted house and there's doors, and he's going to open up each door to the haunted house and describe what's behind door number 1, door number 2, door number 3. This is not just invasive species mussels, and it's not just invasive species milfoil, but there's a lot more going on here. Obviously, and let me just mention the phosphate thing. So I see a guy that runs a spraying business, spraying a lawn of a person that lives on the lake, and I went over and talked to him. I said, hey, do you have phosphates in that? And they go, yeah. Really? Well, what are the rules that you follow when you're spraying around the lake? He goes, oh yeah, we have good rules. We don't spray within a half a foot of the lake. So, hey, leaders of the community that I'm looking at right here, right now, today we're not doing anything about that. Nothing. But at the end of the day, we'll all have an understanding, you know, that's not right. We should do something about that. There's a lot of difference that we can make. There's a lot of ways that you can leave your mark that are not happening today. And I've given you half a dozen examples so far. I'll give you plenty more. There's plenty more that we can do. And it's up to us to expand this to the rest of the community. So our legacy is, it's been easy in the past to just sit here and use the lake. But now, because as Tyler will explain, we're being invaded, we're the last best place, you guys. We're it, and it's coming. And we do nothing or we do something. And so you, by coming here, have said, I want to make a difference. I'm going to do something. So to that end, I'd like to introduce Tyler Tappenbeck, scientist, expert. During his presentation, after his presentation, ask as many questions as you want, but get to the point where you have a good understanding of all the threats we face when he's done, okay? Don't let him leave here until you have those questions. Sherry.
Am I doing the thing first? Or?
But before, as Sherry said, the principal always, I've been called to the principal's office because I got it wrong. The next, sorry Tyler, what we're going to talk about now is something unique to the Flathead, unique to probably, just probably plain unique, and that is, we came up with an idea that we researched grants, and we found one that Whitefish had gotten, and Mike Kupel gave me their grant proposal for 2 years ago. Whitefish got $15,000 from the DNRC for this particular grant. We found that you need to partner with a government agency or a municipality or something to submit this grant. So we thought, hey, the school. So we took Kupel's his grant application, and John Babcock put it into our words. So of the money that I allocated, we were number 5. We were in the top 5 of approval for $10,000. An education program with the school, partnering with the school for invasive species. Mike Kupel didn't get one this year. So we kind of scooped him. I kind of feel bad about that, but we scooped his money for anyway. But Sherry's going to tell us about that program.
Thank you, Dan. So my name is Sherry Stovey. I'm the principal at Marion School across the street. It's a K-8 school, for those of you who aren't familiar with it. We have about 130 students currently. And one thing about our school that I'm very proud of is our science program. Our kids, when you look at just test scores, tend to score above area schools, even the larger schools, particularly in our middle school. Our middle school science program is very strong. We come out very high in our scores and just what they can do. So when Dan approached me about this grant possibility, of course, being a principal at a small rural school, we're always looking for funding opportunities because we're limited in our resources. And so we can't always do the types of things that some of these bigger schools do. And so we take what we have and we try to do the best we can with it. And I think we do a pretty darn good job. But we're always looking for, additional funding resources to improve what we're doing. So I was looking at if you look at our school mission statement, it's striving for success through responsibility, teamwork, great attitude, and respect. And I thought about this grant and I went, that fits right in with our mission. We want our students to be successful, not just in school, but after school with whatever they choose to do in their careers and in life, and in particular to be really strong community members and to be able to be productive citizens. I think we all want that for our kids and our youth. And so responsibility for the lake is just as much a part of these kids' future as it is our future. So, in fact, maybe even more so important to make sure that the kids have a great understanding about the lake ecology and what they can do to protect and preserve it, because they're the future generations. And so, not just kids in our school, but other kids in the community and the surrounding communities. We're hoping to expand this out so that it becomes sort of a valley-wide, county-wide type of thing. And so we were talking about, in writing the grant, we were talking about, well, how can this benefit the Lake Association and the school so that it's a partnership? Because, yes, they needed our organization to write the grant, but part of their, the thing that they're trying to do is educate people and bring awareness, and what better way to do that than to partner with the school. So it works well for their mission and our mission. So we decided that there would be 3 immediate benefits with this $10,000. A lot of it's going to go toward the efforts that the Lake Association is doing outside of the school, but how it will specifically benefit the school is it will give us opportunities to have field trip money. Currently, we only fund 1 field trip, and that's to the Kyla Track meet out of our general fund. That's pretty much all we can afford. The other field trips we do are grant-funded types of things. Through a program called REAP, or we do fundraising or things like that. So this will give us an opportunity to get out to lakes and learn about lake ecology. Specifically, we want to take advantage of the Flathead Lake Biological Station and their K-12 programs that they have. And so one of the goals will be to get as many of our students as possible down to the lake to participate in those experiences. So that's going to be one thing. Second thing is to get some materials in the hands of our kids and our teachers to be able to enhance our science program and our water ecology learning. And so hands-on things where they can actually do experiments with water and with pH balance and all those kinds of things. We don't have very many of those kinds of resources. Books and other materials, videos. Dan has a vision of a section of our library being a lake section, so that would be open to the public as well as our school kids. Sorry, this microphone. Speaking of funding, we need a new microphone system. Anyway, so a section of the library with materials for our classrooms. That's a second benefit. And third, the greater goal is to be able to have our kids become sort of experts on the lake and on invasive species. And then they can take that expertise out to things like science fairs and other, things that are going on in the county, possibly teach other schools about these things and just kind of spread it. Grant funding is an interesting thing. You never know when you're going to have it and when you're not. And so when you have it, you need to make sure that you're using it well, using it to the greatest benefit of everyone as possible. For example, and sadly, we didn't get our 21st Century Schools grant for this coming year, and that is the grant that has been funding our after-school program. So unfortunately, we just found out late last week that we partner with 5 other schools to try to get that grant, and the state of Montana decided that the way to improve the quality of after-school programs was to fund fewer of them in this grant cycle and give them, each of the ones that they did fund, more money so that they can show that they're quality programs. And I understand that, I do, but unfortunately it left a lot of us without funding, and a lot of our parents and kids, I'm getting off on a tangent, sorry, but a lot of our parents and kids depend on that program. And so we're trying to now look at how can we raise funds, how can we support the after-school program. So getting back to this grant, this is something we do have and we can use, but it's just for 1 year. And then after that, we'll reapply and hopefully get it again, possibly increase it. If we don't use it well this year and don't do some great things with it, will they give it to us again? Probably not. So we're trying to work really closely as a partnership with the Lake Association, with Project WET, with all of the experts, Fish, Wildlife, and Parks, all the people who are willing to work with us to make sure that we're using the grant wisely and to benefit everyone, the school, the community, and the lake. And so with that, I'm going to pass it off to our school board chair, Lily Brower. She has just a few words as well, and then we'll get back to our keynote. Thank you.
Thank you, Sherry. The school board they're just really excited about this whole grant process with partnering with the lake. And I'm feeling that if the kids learn early to take care of our water, to take care of our lake, to be responsible, they'll pass that on as adults and then to their family members and to other people. So again, Sherry pretty much covered, everything that the school is involved with it. Our sixth graders do a is that three-day?
Yeah.
3-day camp on the lake in May every year. And so again, our science teacher is really wrapped up in a lot of that kind of stuff. And so this program would help them understand more of what goes on in the lake and what needs to go on in the lake to keep it pristine as it is. Oh, thank you. Danny?
Thanks. And now Tyler Tappenbeck, Flathead Biological Station, University of Montana scientist. Tyler.
Thank you, Dan, and thank you everybody for coming out today. I know it's hard to take time out of your busy lives to come listen to some guy talk in front of you. Like Dan said, I'm with the biological station. I've been there since 2004 as a research scientist and a graduate student, and I've worked on Flathead Lake for the past 10 years. And so I'm here to talk to you about just general lake ecology and, like Dan commented to, the house of horrors of the threats that your beautiful lake faces. And so first, when we think about lakes, lake ecosystems, we have to really acknowledge that lakes are really the compilation of living and non-living things, plants, animals, microorganisms, and how they interact with the physical and chemical environments. And that it's not just within the lake, but at the landscape sort of level. And so to really understand lakes and thinking about lake ecology, we have to think about how lakes are structured. And we can do that by identifying zones, not only in depth, but in distance from the shoreline. And so when we think about zones of a lake in depth, we have the photic zone, which is where light penetrates. We have the aphotic or profundal zone, which area where light does not penetrate would be the deep areas here, and the shallow areas here. And then we have the benthic zone, which is sort of the bottom of the lake. And then when we think about lake zones as far as their distance from the shoreline, we can think of the littoral zone, which would be this shallow area here. And then as we move up in the deeper open water, that would encompass the photic zone, the profundal zone, and also the benthic zone, we have the limnetic or the pelagic zone. And so the littoral zone is comprised of its warmer, shallow shorelines around your lake. Can have a diverse community of organisms, periphyton, which is just the algae that grows on the rocks here, snails, amphibians, insects, fishes, and so lots of different organisms live in the littoral zone. And subsequently, it has a very close connection to the terrestrial landscape and terrestrial food webs. And as if we think about the pelagic or the limnetic zone, the big open water area, We're mostly dominated by the small crustaceans and zooplankton here, algae, and then microbial communities, as well as fishes. And then we have the deep water zone, or the aphotic zone. This is the area that light does not penetrate. And this is a good area for mysis shrimp. They like to hang down on the bottom. You find many composers and decomposers here. And subsequently, whoops. This is also the area when the lake stratifies. And when we talk about stratification, we have different things that occur in the lake during different times of the year. And so in the summertime, you might notice when you jump into the lake, it's very warm. But if you were to go down deeper into that lake, it'd be very cold. And so we have what's called summer stratification here. So in the summertime, we have 3 different areas of lake. The epilimnion, which would be the upper layer, the metalimnion, which is the middle layer, and the hypolimnion, which is the lower layer. And all these areas have different temperatures. And the stratification occurs because there's a temperature gradient between these areas. The thermocline occurs right here in the metalimnion. And essentially that is, as you move down through the water column, you'll find an area where you have very sharp changes in your temperature. And subsequently, water is most dense at 4 degrees C. And so as your water becomes less dense, warmer, you can have lots of cycling that goes on, or movement, but as wind comes into the system, it will not penetrate this thermocline and mix up the bottom of the lake. And so your organisms, as your communities are growing, your algal communities are growing through the nutrients in that system, they're going to deplete that upper water column, and a lot of your nutrients will stay down this lower water column, and the upper water column will never see those nutrients. And subsequently, you may not have organisms that are able to penetrate this thermocline and then settle down the bottom. And so it stratifies, it prevents the lake from mixing completely. Whereas opposed to in the fall and in the spring, your systems are pretty much, if they're not frozen over, they're the same temperature top to bottom. And so they very easily mix. And so you get this mixing. And so you have this cycle of what your lake is doing now. So we're in the spring now, we're turning over. And then we're starting probably to set up in stratification. And so that has implications on not only the communities that you'll find, the nutrient cycling that's going on, and how your lake is set up and functions. And so if we look at the benthic zone, could be either the bottom of the lake in the shallow littoral zone, or the bottom of the lake in the limnetic zone, and this is where you find the bottom dwellers. You find your crustaceans, your worms, your protozoans, insects, microbes, and some fishes. And so the point of all this is we have to think about lakes as not just a big bathtub. They have different zones, but all these zones interact, right? And so if we think about the food web in Little Bitterroot Lake, we have kokanee salmon. Kokanee salmon feed on zooplankton out here in the pelagic or the limnetic zone. Those zooplankton feed on algae. Those algae feed on the nutrients that come in from the rivers or deposited via aerosol deposition. And then in the littoral zone, we may have your rainbow trout. And when they're young, they might be out here feeding on snails or crustaceans that feed on the aquatic vegetation or the algae that grow on the rocks. But through time, they will move between these different areas and up and below into the water column. And so these organisms and nutrients move throughout the water column and not only vertically but laterally throughout the system. And so it's very important because it's not just a bathtub. All these things are interacting depending on time and scale, and we need to think about that because healthy food webs need healthy water. And so we have to protect that in order to protect our food webs. And so what influences these food webs? I mentioned a little bit in that diagram there, but temperature. Different organisms need different temperature regimes. We have very cold water here in Little Bitterroot. That's why we can support kokanee salmon. That's why we can support trouts. Algae need light on top of nutrients to photosynthesize and to produce. And so that's a very important key feature. We need to understand the water clarity. And so Water clarity indicates water quality often. It's one of the oldest measures of water quality. And so understanding the water clarity is going to tell us how well light is penetrating down into the water column so that the algae can obtain it to photosynthesize. We need to know the dissolved oxygen in the water and how that plays in the food web, because fish need oxygen, organisms need oxygen to live. We need to understand the pH of the water column, or how alkaline or how acidic it is, because that's going to play not only in chemical and biological processes, but it's going to influence whether organisms can live there. And we need to know what the nutrient contents are. You know, what the nitrogen and the phosphorus are, the aquatic fertilizers, because that's going to play into not only microbial processes, but also it's going to play into our algal communities, how much they produce. And also whether that nutrient content is enough to keep the lake healthy or send it towards a state that may be not healthy. We need to know the organic carbon. Carbon is used in many biological processes, from reproduction and photosynthesis to the creation of proteins and lipids and sugars that organisms need. And the cations and anions, these would be the trace elements, so sodium, calcium, things that organisms need to make different compounds for life. And then we need to understand the stream runoff and how much not only sediments, but nutrients are being deposited in our system and how that might influence lake food webs. And then also atmospheric deposition, what's coming in off the rain, fires, what's bringing nutrients and sediments into the system. And then understanding those predator-prey relationships. Understanding that, you know, what are the different food chains and how do they influence the food web.
So this slide is important, it needs some mention in that the gentleman over there, Mr. Babcock, is going to talk about what the Little Bit Lake Association for many, many years has been studying. And you'll see these again: temperature, light, water clarity, dissolved oxygen, nutrients. So we're going to come back and talk about what is actually the measurements in our lake of those things, which will tell us the health. He's sort of like the doctor of the lake. He's given the lake a physical for the last 9 years. He's done it for the last 19. Little Bit of Lake Association. So when we're done here, I know this sounds like a chemistry experiment and all, but this is important to build a base for our understanding of what really is going on there. So please be patient with the detail here, but realize you're going to see this again, but you're going to see it with regard to the measurement that we have taken for many years of our lake and where it's at. We'll come back to this again, these items.
And I'll come back to it a little bit here as we move through. And so how do we measure these things? Right? John's going to talk a lot more about this, but we can use water bottles that we send down to different depths in the lake. We collect water discreetly, we fill up bottles, we bring those bottles back to the lab. We analyze for all those different things that I talked about: nutrients, cations and anions. We take out units that have multi-sensors on there that measure dissolved oxygen and pH and temperature of the water. We send down light meters that measure the amount of light that's penetrating down to the water column that's available to algae. And then we use a Secchi disc, which is one of the oldest limnological instruments around, that we lower that down and we can determine the water quality. And so why should we care? So this is getting back, I told you about all these things that we measure, all these things that are important. Well, because then we build long-term data sets. This is some of John's work from 2004. It's essentially showing that nitrogen and phosphorus in the lake is on a downward trend, which is a good thing. And then if we look at the volunteer monitoring project, not to get too specific here, but all of these measures, so this is the chlorophyll content, or the amount of pigment that designates photosynthesis in algae, the total nitrogen and total phosphorus, and the Secchi depth, so that round disc that I just showed you. And essentially we have formulas that we can calculate as trophic status, and that tells us that it's oligotrophic. So it's nutrient poor, it's good water quality. We also have years of data that show us that it's very cold water in this lake, which is good, top to bottom. But that, like I talked about earlier, that the lake does stratify in the summer. We also see that this lake has high oxygen. And that, like I stated, you know, we're starting to see a drop in nutrients, which is good.
On mussels with our lake the way it is now, will they survive?
We're going to get there.
Okay.
All right. So, when we measure all these things, and what we know now is the lake is in good shape, and it's been that way for a long time, but there are very, very serious threats, and there's cause for concern. Number one would be non-native species introductions. Now, when I say non-native species, I say that something that is not endemic here and that can be brought in to potentially alter the food web or be of detriment to the water quality of your lake. Now, Little Bitterroot Lake has introduced species. Rainbow trout, kokanee are introduced species, but it is in a balance. We have good water quality. This is a healthy lake. And so when I talk about that, I'm talking about new things that come into the lake to change the current state of the lake. And then we're going to talk about the threat of nutrient pollution. And thirdly, we're going to talk about toxic chemicals and biological pollution. And so when we talk about non-native species introductions, we need to first talk about something that's close to home for me, working on Flathead Lake, mysis shrimp introductions. Mysis shrimp was introduced to Little Bitterroot Lake, Swan Lake, Whitefish Lake, Dickey Lake, and various other lakes within the basin. It was not introduced to Flathead Lake, but it made its way in, probably from Whitefish and Swan Lake. And subsequently, it had some pretty detrimental effects in Flathead Lake. And so this has a lot going on here, but just what's important to note is that this is the abundance of organisms, and this is time, and this is our kokanee populations up until the introduction of mysis shrimp. Mysis shrimp were first found in the mid-'80s in Flathead Lake, and then their population skyrocketed. They're in magenta here, and so you can see them through time. The green is bull trout, and the yellow is lake trout. And so with the introduction of mysis shrimp, it provided a food resource for lake trout. Lake trout were in the lake since 1910. They co-evolved together in Canada and other lakes. And so once this food resource was provided to lake trout, plus kokanee as a food resource for them as they got bigger, we saw this drastic increase in lake trout populations. Mysis took over, and bull trout subsequently were impacted not only through predation but competition. And so as I talked about, the reason that mysis shrimp were introduced to some of these lakes throughout the basin is that they provided a food resource for kokanee. That was the state's goal. And in some lakes, that worked out great. But in Flathead Lake, it did not work out so great. And when the mysis shrimp were introduced, they started feeding on the zooplankton, which were the primary food resource for kokanee. And so we see that there was a dramatic drop in abundance and a community shift with the increase of mysis, because they basically came in, ate the zooplankton, changed the food resources for kokanee on top of predation, and crashed the kokanee population. And so this figure shows that, before the introduction, we saw a lot more native fishes in our samples. And then after the introduction of mysis, we saw a drastic change in the increase in exotic or non-native species. And so when we talk about non-native and introduced species, of course we got to talk about it. It's amazing to me that there's still bucket biology going on, but there is. And so recently, as late as 2014 or 2015, we're seeing continual introductions, illegal introductions of non-native fish and potentially harmful fish to lake food webs. All of these fish are piscivorous, means that they eat other fish, and they're very good at it. And so think to yourself, what would happen if a northern pike got into my lake and started reproducing and became a good population? They'd probably feed on all the salmonids and the other fishes in the lake. and probably have a major impact on the fish community in here. And as a subsequent, those fish that I just showed, northern pike, walleye, and bass, really like the habitats that aquatic invasive plants provide. They're mostly sit and stalk predators, and so these plants can provide better habitat for them when they invade a lake. And so we don't want those to come in either. But aquatic invasive plants are a real threat. And right here we have 4 major ones that we need to be concerned about. We have the fragrant water lily here. We have the curly leaf pondweed here. We have the flowering rush here. And we have the Eurasian milfoil here. And so these non-native plants were native to Europe, Asia, Australia, or Africa. They grow along shorelines and slow-moving areas, very dense. They outcompete the native species. They can provide habitat for snails that cause swimmer's itch. If you ever had that, that's not very fun. And like I mentioned, they provide habitat for invasive species, fishes, and that they overwinter and grow very rapidly in the spring, which can cause the littoral zone or the shorelines of lakes to look like this. We don't want that. And they spread very easily by small fragments of material. And so this map shows from 2016 the distribution of all these non-native aquatic plants. You'll see that the curly leaf pondweed is the most distributed, followed by the Eurasian water milfoil, and then flowering rush, which is, we've got it all over, especially on the south end of Flathead Lake. And I want you to notice something here. There's nothing in Little Bitterroot Lake, as we mentioned before, but it's all around you. And so that's a real threat. And so what's waiting on the doorstep here, right? We're going through all these different things that are potential threats. Well, you got it. It's the next Flathead Lake monster, the real Flathead Lake monster, all right? We're talking about zebra and quagga mussels. Zebra and quagga mussels were introduced in 1988. They're native to Eurasia. We believe that they came on the ballast water from ships, and they were first found in Lake Erie. Oops. 10 years later, they're all over the Northeast and the Great Lakes, spreading down south and through the Midwest. By 2005, note this line is the 100th meridian. It designates east versus west. They're making their westward movement, primarily from transport on boats or other industrial means, boats that are being transported, or machinery. By 2008, they've crossed and they're into the west. And by 2016, as we know, they're here in our state. Suspect samples were found in Candy Ferry Reservoir, but we've, through eDNA or DNA detections and microscopy, determined that they are in Tiber Reservoir. And so this is why the threat is so real, folks. These green dots are boats, and these lines designate their travel distance to Montana. They're coming right through where it's all started. And this is why we're so concerned. This is what they do. They clog up boats, they litter shorelines with their shells and overtake it. This is a shopping cart that someone pulled out of a lake. Any strut, they can literally attach to almost anything. They're very small, max of 2 inches in length. They can attach to almost anything, like I noted. They reproduce in cold water, like we have here in Little Bitterroot Lake. There can be up to 10,000 per square foot. Think about that. An adult female makes more than 1 million eggs in her lifetime. And they're filter feeders. They can filter up to 1 liter a day, or a quarter gallon. That's just one mussel. That's not the 10,000, that's one mussel. They're very robust. They can survive out of water for many days, and the larvae can survive in water for weeks. And once they get widely established in a lake, there's been no eradication. And so, if we think about the ecological effects, they're filter feeders. They pull small organisms, algae, particulates out of the water column. And so they can remove up to 80% of the edible plankton, which are not available now to zooplankton or juvenile fishes. They can disrupt the entire food web in that respect, and in turn cause fish populations to crash. They can promote toxic algal blooms, like this. And then, like I showed on that other example, they can cover all the available substrate, like this. And so that's, you know, an idea of what could happen ecologically. Now let's talk about us as humans. What do they do to us? Well, they clog intake pipes. If you draw your water for irrigation, for drinking water, they'll clog up your pipes. And here's where it hits the dollar. Greater than $3 billion was spent by the Great Lakes in the 1990s due to mussel invasion. They estimate that greater than 90 million could be spent if they get into the Columbia River Basin. And over 5 billion estimated for the Pacific Northwest. How would you like that to be your job? This is a gentleman having to pressure wash the inside of intakes for hydroelectric dams. And so this cost, because of all of the extra money that's going to go in for power companies, for Kerr Dam, for all of the places that we obtain our energy from, that cost in our energy is going to go up. We're going to have decreased recreation and angling opportunities, and also the economic issues that go along with it if you're a guide, if that's your livelihood. And so we'll see fish population crashes. Water bodies will just simply have to be closed because we don't want people to be able to transport these things. And these beaches will be unusable because they're covered in shells, like the photo I showed you earlier. And so there hasn't been a good economic evaluation of what's going to happen to people's property values, but I think you can get an idea that you know, if we take what has been looked at in other AIS, it's been shown to be a 20% drop in property values. And we have an economist at the biological station, his name is Nanette Nelson, who's got a grant to look at the economic impacts in the Flathead Basin for the introduction of quagga and zebra mussels. And so the point here is that this is avoidable through public interaction, early detection and monitoring, and the clean, drain, and dry programs, that this does not have to be Little Bitterroot's future. And so the next thread I would like to talk about would be nutrient pollution. Well, what are nutrients? Nutrients are just aquatic fertilizers. In general, we have very clean water quality here in northwest Montana. Phosphorus is linked to the sediments. And because of that, we have very low P because of the geology of where we live. And as a consequence, many waters are P limited, meaning that for a lot of the biological processes that you need nitrogen and phosphorus. And so we call them limiting nutrients because one limits the other. You need them both to perform a lot of necessary biological functions that we see in food webs, especially with growing algae. And the nitrogen is linked to biological cycling. It's an increasing trends of concern, as we'll see shortly. Essentially, living things need nitrogen and phosphorus to grow, but it's when we have an excess that we get toxic or detrimental algal blooms and decreases in water quality. And so when we have those excess nutrients, we can get things that not only are toxic to our pets, but also to humans. Oop. Sorry. And so phosphorus was a historical problem in the basin. With older wastewater treatment plants, failing septic systems, and a lot of detergents, whether it be hand soap or dishwashing detergents, contained phosphorus. The good news is the Clean Water Act put restrictions on the amount of phosphorus that could be released into fresh water. In the 70s and 80s, we had upgrades to our sewage treatment plants, and in fact, we had some of the highest technological sewage treatment plants for the time. And we got a ban on phosphorus in detergents, and that occurred in Flathead, Lake, and Missoula counties. So that was a big win. And so as a subsequent, we see that the total phosphorus, and these are from sewage treatment plants around the valley, dropped through time. However, the bad news is nitrogen went up. And so some data from Flathead Lake, and this is, the colors just denote different depths in Flathead Lake. Through time, we're seeing an increase in nitrogen. And this is not just a phenomenon that's occurring in Flathead Lake. Globally, we're seeing an increase in nitrogen. So there's the current problem. We're seeing this increase in nitrogen. Septic systems are outdated. EPA recommends that they're replaced every 20 years. Not something that financially people can afford. And sewage treatment plants are outdated. They need upgrades. They got the phosphorus figured out. They didn't get the nitrogen figured out. We're seeing an increase in atmospheric loading. So forest fires provide nitrogen and phosphorus. Population is increasing. So a lot of people, you have a lot of nitrogen. Land use changes. And we're seeing an increase in loading in our tributaries, potentially from the use of not only fertilizers, but animal waste. And so again, you know, algal blooms that cause fish kills, algal blooms that line our shorelines are avoidable in here. You know, do what we can. Minimize the use of fertilizers, use best land management practices. And the big thing is to keep the phosphorus out. Little River Lake actually has some nitrogen in it, and keeping the P out will keep those toxic algal blooms from occurring. And so lastly, I'd like to talk to you about toxic chemicals and biological pollution. And so the current problem is that we're starting to see fecal coliforms show up in stormwater. So fecal coliforms are bacteria that occur in the guts of animals, and if we ingest them, even though animals have them, then we can get sick from it. And so the EPA designates that 0 bacteria in 100 milliliters of water is safer. So nothing, no detection. For swimming, it's 200. We have reports of there being counts of 620 in Kalispell and over 7,000 at a dock in Big Fork. So they're getting into our surface waters. Little less talked about would be these semi-volatile and volatile organic compounds and these chemicals. And these are things that can cause not only are carcinogenic, but also can cause issues with our central nervous system. And then we have the personal care products, the things that we use every day. Our body doesn't use all of the things that we take, and so we excrete it. And so those are winding up in the surface in the groundwater, things like acetaminophen, warfarin, which is a blood thinner. And so in 2010 and 2011, the Biological Station conducted a shallow groundwater study to look for these things throughout the shallow alluvial aquifer of the Flathead River. And we selected 31 sites, which was a combination of not only monitoring sites that had been put in the 80s, but also we put out public service announcements and we asked the public if they'd want to participate in the study by allowing us to sample their drinking water wells. And these wells were 18 to 31 feet. So how many people in the audience have a drinking water well, and that's how they obtain their water from the, right? Who has a well that's deeper than 30 feet? Who has one that's shallower than 30 feet? So we were more concerned about these shallow wells because we felt that they would be the most susceptible to contamination and pollution. And so when you show up at someone's house dressed in a suit like that, you get some weird looks and people get very nervous. But that's mostly so that I don't contaminate things. We're looking at some of these pharmaceuticals and personal care products down to the parts per trillion. So very low compared to what a doctor would prescribe you or you would take over-the-counter medicine. But we don't want it to contaminate it, so. And here's what we found. Don't get, tied down with the numbers. Just know that some of these things are plasticizers. They're things that are carcinogenic to humans. And they're found in herbicides, pesticides, things that we use and don't really think about them infiltrating into the groundwater. And so those were detected in 17 of the 31 wells that we analyzed. And the VOC, so these are the things that volatilize quicker, so a little bit harder to find in the environment. But still, we found these xylenes, which are carcinogenic. Chloroform, And lastly, the pharmaceuticals and personal care products. Now, like I said, these are found in parts with very, very low levels as far as toxicity to humans. But think about if you're a small zooplankton, very small, microscopic. That might have a pretty big importance in how it moves through you and how it influences you. And so we detected them in nearly half of the wells that we sampled. And so septic systems and sewage treatment plants just aren't designed to deal with some of these things. And so there's no way to biologically remove them. It's a real bummer that we're no longer able to recycle plastic in the valley. And so some of those plasticizers that we found are going to potentially find their way into the groundwater and into our lakes and our streams more so than they were before. And we just don't have a good handle on how these compounds affect food webs in rivers and lakes. I mean, there's evidence on some of those hormones affecting fish and altering them, but as far as how they move up the food chain from microbial community to the algal community to the zooplankton community to the fish, we don't have a good understanding. But there's hope. We have a graduate student that's working on that in Flathead Lake to see if he can detect these things in the organisms in the water in the lake. And so, to be a broken record, these things are avoidable. I think that through public effort and properly disposing PPCPs, don't flush it down the toilet. That's just going to be even more than what we excrete as far as going into the septic systems. I think that we can help minimize these harmful chemicals. And with that, I appreciate your time, and if you have any questions, I'd love to take them.
I'm gonna rephrase my question.
Yeah.
All right, with knowing the mussels
Mhm.
in our lake, the nitrogen, rock source, whatever, can a mussel live in our lake?
Yes. Yes. There are literature that put these sort of designations of what is favorable conditions for a mussel based on the calcium content, based on the pH of the water, right? And so the few samples that I have collected, the calcium range, or the average range is around 12. That's on the sort of unavoidable sort of range. But I would not take that as end-all be-all. We don't know. These are other studies from other places. They haven't been to our systems. And so we can't say as a matter of fact that if they got in here, the invasion would fail.
And Lauren, that's one thing we added this year to our program. There has been some sampling in the past for calcium and hardness and alkalinity. And mussels really need those to help build their shells. And we haven't really sampled for that on an annual basis, but that's something we added back in this year to kind of support the AIS side of things so that we can get a better look around the lake, like where there might be parts of the lake that have higher levels of calcium than others. You might have low calcium in the middle of the lake, but if there's an upwelling of groundwater on a certain side of the lake, that might be more favorable to mussels. So this year we did add certain parameters that are going to kind of indicate potential colonization.
And what is that test called?
Calcium is probably the biggest one because that's what they build their shells out of. So, but yeah, pH, calcium, hardness, they're all kind of relate to each other, but we added those in this year just to try to kind of beef up the AIS portion of our sampling. So.
What's happening on a research basis, biologically, genetically, to combat mussels?
Well, there are mussel sites that are being used, and not in Montana, but in other areas of the country, to see if if they have a lake where a new invasion has occurred, if that can work. And let me go back here in a couple of slides. There are a few places where they have successfully eradicated mussels, not very many. And if we want to go, let me go to the slide, I can show you this. These purple squares here are the only places that a successful eradication has occurred. And it would be through those means. Getting a little bit more sci-fi, for other things, they're talking about trying to figure out genetically a way to create a virus or something that may attack the mussels, but that's way off.
Do you know what they use? Did they use a chemical to kill those mussels? Or?
Yeah, it was a mussel-side, something that was developed to kill them, yeah. And that would probably, I bet it would be a small lake, early invasion, something like that.
At least in the instance of the one in Virginia, I know that was a quarry that they basically reset. They killed everything in it to kill the mussels. So that's not an option that we would have for open water.
And something I didn't talk too much about in the biology of the mussels is that, that female puts out millions of eggs, the males put out millions of sperm. Once a juvenile is created, a veliger is the juvenile or the larval form of a mussel, it's out in the water column for months until it finally is able to attach to something, and then it starts up an existing population in a certain area. So the ability to kind of, once we detect these things, they could be everywhere. And so think about trying to use a mussel side in Little Bitterroot Lake or Flathead Lake to address things that might be everywhere. But we just might not have an adult population yet.
Can you comment on least impactful ways of weed control and the impact of the control chemicals and things that are in the lake?
So that would be on the landscape?
Well,
Or if we got them here?
water, getting in the water table,
Yeah. We live on a creek, and I'm hand pulling everything that's even within 200 yards. And that's just me. The connectivity of the shallow aquifer, even in a small creek, even our creek it's seasonal, but it takes a little bit of rain for things to in that water table to reconnect. And so, unless you know the groundwater influence and the flow paths, I would say if you can, don't use any herbicides. Just use hand pulling if it's feasible, or spot spraying. But broad applications would be minimal.
How are you gonna control these noxious weeds that my property is infested with? You just bought with. the whole neighborhood where we live is infested with natural weeds. How are you gonna get rid of these things?
Well, I mean,
Hard work.
very hard work. I mean, physical. I mean, so weed removal is not just herbicides. It's a combination of multiple methods, and physical removal as well as some treatment application. But if you're anywhere near a water body, I would suggest that if you're going to spray, you spot spray. Do not do broad applications. If you have an area where there's a high concentration, maybe you focus there. But if you can eliminate spraying close to the water, I would recommend you do that.
Do you have a product in mind that you would recommend for spot spraying?
I cannot do that.
Yes.
Back to the mussels. Do you know of any research being done to turn these into a food source? We can eliminate a lot of species just by eating them.
Yeah, no. I don't know of any tries on that. We joked around and thought about doing that with Mice's Shrimp in Flathead Lake, see if we could fund some research that way. But no, I don't know of any methods to try to do that. And they're, you know, I mean, they can get relatively big, I guess. If you had somewhere on the bigger end of the size range in the 2 inches, maybe that would be something you might want to try to eat. But if they're really tiny like that, it would take a lot.
Just remember,
Yeah, or I guess
they're small, highly concentrated
Yeah.
filter feeders. We don't want the heavy metals and things and that's that
Yeah,
they,
and
they
that's
have in their
right.
That's,
That's a good point. Is that when they filter out all of these small particulates and organisms out of the water, they bioaccumulate and they concentrate toxins. And then when they excrete those, so the areas that these mussels live are often high in pollutants, high in toxic chemicals. Anybody else?
How long does the herbicide stay active if you spray 1 year? And then the other thing is things like DEET. How long are they still a problem? Is it multiple years or?
That's a very good question. I don't know the half-life on some of the chemicals for spraying or DEET, but for example, when we sampled these and got these detections, these were winter and spring samples, and so it could have even been months ago that it got into the water columns through someone washing it off, right? So if we're detecting these in the off season, then it could persist for months. But I can't really speak to how long it's staying in there. There's been no studies, just any sort of tracer studies that I know of that have tried to put in a slug of DEET or some of these toxic chemicals and see how long they persist in the groundwater.
Are boats or recreational vehicles the only way that these mussels or plant life can be transmitted from an area to another?
That's a good question. No, they can attach them. So correct me if I'm wrong, that felt-so shoes have been outlawed in Montana. Is that correct?
no, they have.
No, they haven't. Okay, so I had heard, that was a rumor I had heard. So a big problem is felt-sewed wading shoes can collect material or could collect the veligers, and because it's that felt, it stays wet for a long time. So if you did not completely dry your fishing equipment, you could transport it that way. Your boats have been the primary vector for moving these things around, but your fishing equipment could be a way if they don't get cleaned and dried. I'm trying to think of anything else that might be.
Or the migratory birds?
It's possible. I don't know of any studies that have looked at that. We're doing some work in Glacier National Park, and we're looking at some of the food webs up there, and we're finding zooplankton in new lakes that are less than 50 years old, and there's really no way that they would get in there other than if they were transported by birds. That's one of the big things, or wind, or things like that. So it's very possible that the veligers could come, be in the water, a duck lands in the water. If it's wet and its feathers as it flies off to another place, it could be. But I don't know of any studies that have looked at that. That's a great question.
For organized prevention for boat check stations and things like that, will there be some sort of government or academic money released, or will it need to be legislated? Do people need to get this on the ballot to spend tax dollars to set up check stations, or what do you recommend?
Well, I think Russ could definitely address how that's being funded through the state, but I'll talk about the biostation a little bit. On Flathead Lake, we have legislative money that we use in our monitoring program to monitor the water quality in the lake. Through donations and our legislative money, we have enacted a EDNA, an early detection monitoring program for quagga and zebra mussels. And so a combination of philanthropy, state legislative money, we have been able to put that in place on our lake. And so folks that may be able to contribute to some sort of fund or an endowment to put that in place would be one way. Absolutely address the legislature. I know that the state's doing all that they can with the money that they have available, but yeah, that's a hard question, because everybody wants this stuff, but we got to get the money. Where's the money going to come from, right? And so we're scraping together whatever we can to be able to have a monitoring program. Writing grants, another option. Writing grants to be able to sample 3 times a year on Flathead Lake. And so we're primarily focusing on the eDNA techniques. And those of you that don't know what eDNA techniques are, is that we can go out in the water, whether we take a whole water sample, or if we run a net through the water and we collect all that material, we take that material back in the lab and we look for the DNA, very specific DNA of the mussels. And so that's how we feel we can cover a lot of area. We can take care of we can process a lot of samples to be able to look for it. We complement that with what the state is doing on the microscopy end, where they're looking for the juveniles through a technique called cross-polarization. And so these veligers, they fluoresce, their shells fluoresce. And so when you're looking through a sample under the microscope, you can see these organisms. And so those two techniques complementing each other are our best hope for early detection. Prevention is by far what we have to do. But using these early detection techniques, at least we'll know. And then we can put into place practices that say, this lake is infected, we need to isolate it. Try to eradicate if we can using all the available techniques that we have at the time, but we don't want it to go somewhere else. So not knowing is the biggest issue. And so we need to prevent and have these detection techniques in place so that hopefully we can stop it from going somewhere else and contain it. Yes, sir.
I know that later in the season when the water levels are down, you're backing farther in, especially on the shallower ramps. Has there been any studies done about the vehicles launching the boats that are going into the water?
That's a good question. I have not looked at any specific details on that, but I would say that, you know, for one, the boat, if it was used in the lake that was infested and it remained in that lake, that's where the adults may form onto the boat, right? And so you get launched, and then if it's a time during spawning, they're going to release things into the environment. Their eggs and their sperm create the veligers, and the veligers go to populate the lake. The other thing would be, is there water on the boat? So travel time, unless it's right up the road, I would think would be long enough to dry out the vehicle, unless there was somewhere they could hold water that had veligers in it, the juveniles, and then backed into another lake, and then release that water and the veligers into the lake. So it's areas that can hold water, keep things damp so that the veligers can live, or that have the mussels physically on them. And so the mussels, the adults can live out of water, they close up their shells. The veligers can live in the water, ballast water, your boat, your bilge water, for weeks.
We haven't seen it
No, yeah, no detection. So something that I didn't mention, which is that University of Montana since 2013 has collected DNA samples, water samples from here, and we have not detected milfoil or zebra mussels.
And is there a number in our testing that we're doing right now that this milfoil won't live in our lake?
That I don't know, John.
Good lake?
I've tried to look at like habitat requirements for milfoil, and it can live in a wide variety of habitats. And our water quality and habitat isn't starkly different from Knoxon Reservoir. And if you go to Knoxon Reservoir, it's everywhere. You know, and I think the efforts that they've done to look for milfoil and these other invasive weeds haven't been very comprehensive. So that's one thing we're trying to do with the grant this year. We'll be back in August, and we're going to do a full shoreline survey. We did the survey on Knoxon Reservoir last year, so we're familiar with how to identify, you know, not only the invasive species but the native ones as well. So we'll kind of put together by the end of the year kind of a baseline map of what is out there. And it's not to say we're going to, you know, look at every square inch of the lake, but I think we're going to do enough of a survey that if it's out there, we'll hopefully detect it. And if you go down to Knoxon, I mean, some of the boat launches, it's so infested with it that, I mean, you launch and you're just trudging through weeds and stuff. And we don't want to see that here. And like he said, he showed that map, we're surrounded by it on all sides. So I think it's a pretty big threat, you know.
Thanks, Tom.